Hello students, this is Faizan Mirza. Here we are discussing mutations today. Mutation could either be a gene mutation or a chromosomal mutation. A chromosomal mutation shows up on a karyotype because the number of chromosomes do not tally with the normal number of chromosomes of that species. A gene mutation is occurring when the person is having a change in the genetic sequence, a change nucleotide sequence, and because of the gene mutation, a new allele will be forming, and that new allele in most cases will be a disordered allele, or it will be coding for a protein which will not be performing a normal function, and in most cases, and it will lead to a disorder. The disorder could either be a recessive disorder, it could either be a dominant disorder. For a recessive disorder in the gene mutation, the person must have both the recessive alleles to have the disorder. But if the disease is carried by a dominant allele, then in that case, even a single copy of that dominant allele can cause the disease to be passed on from one generation to the other. Mutations actually affect the phenotype because of the gene mutation, the new nucleotide sequence will end up producing a messenger RNA, which will be having a set of different codons, and those different codons will end up having a different alignment of amino acids, and the primary structure as well as the tertiary structure of the protein will start to change. So there are three ways primarily in which gene mutation may occur, either by base addition, base deletion, or base substitution. Base addition and base deletion, they both are a type of frame shift mutation. What happens? The entire reading frame changes. For example, in base addition, there are three codons. For example, GAG, CCT, GAG. In this, you see the second codon CCT. Let's suppose the second codon CCT, its first position C, here a T gets added. So when T gets added, the two C's they get pushed ahead, and the third codon changes as well. So what happens? The frame shift occurred because of the insertion of this particular nucleotide at this position. So the reading frame changes altogether. In base deletion, the existing nucleotide sequence will end up having a base deleted. For example, here this bold C, if it gets deleted, so the frame shift will occur towards left. It will come back towards the first codon. So again, the reading frame has changed. You can see that the first nucleotide is very much intact, but the second, third, and the one which will be following these will change. So in base addition and base deletion, which both are frame shift mutations, the protein to be coded is basically highly abnormal because the codons are very much changed. But again, it depends on which codon is basically mutated. For example, if it's a 50 amino acid, 50 codon long genetic sequence, and the base deletion or addition occurred on, for example, the last 48th or 47th this codon there, in that case the effects will not be that deleterious. But yes, if it occurs earlier in the chain, then the entire abnormal protein will be made, or no protein will be made altogether. So it depends on which codon is actually affected, and that will actually tell you how bad the protein will be. Then there's base substitution to be discussed. Base substitution, there's this existing nucleotide sequence. In this, for example, in the second codon, on the second position, instead of a T gets placed. So this is substituted base. A substituted base does not bring about frame shift mutation, but it changes the codon. And because of this change codon, a different amino acid might be coded or might not be coded by this particular change codon. Let's discuss this in detail. What's shown to you here is the appendix or the dictionary that we use for codons to predict the 64 amino acids that can be brought about. The 61 amino acids and the three stop codons that are coded by different codons here. So let's suppose if you have a GGT codon and it codes for pro. How do you know that it codes for pro? You know this one codon GGT. It's having three positions. First position is G, second position is G, the third position is T. So in this table, we go for first position. We go for to the column where G is mentioned. In the second position, again, this is mentioned G. So in second position, we go to G. So these G and these G, they tell us that we have to look at this box. And then the third position is T. So in the third position column, we locate the T, and we come back to this. We see that, okay, this is the pro amino acid, this coded for GGT. So GGT, wherever the codon GGT will be there, will be coding for the amino acid pro. Let's suppose GGT changes to GGA or GGG or GGC. The last position of this codon is changing. So keep changing A, then G, then T, and C. You will see that in all these sequences, the amino acid pro will be coded. So basically, the changing of sequence of GGT to GGT, GGA, GGG, GGC, these are bringing about no change in the amino acid. This is called as a silent mutation, when the altered codon still code for the same amino acid. What is missense mutation? Missense mutation occurs when an altered codon code for a different amino acid. For example, if GGT changed to TGT, it would code for THR. If GGT changed to AGT, it would code for SER. So these are basically different amino acids. This missense mutation. Nonsense mutation occur when an altered codon basically is a stop codon and the polypeptide chain terminates abruptly. For example, the stop codons are ATT, ATC, ACT. These are the only three universally stopped codons. So if these codons are made by either base substitution, base addition, or base deletion, the stop codon will cause the polypeptide chain to terminate prematurely. You have to keep the concept of point mutation in mind. Point mutation basically refers to that the mutations occurring at only single position, which means one out of three on a codon. So one nucleotide is changed because of point mutation. Then because of that point mutation, either a frame shift could occur or frame shift could not occur, depending whether the base substitution, base addition occurred, or was it base deletion. So that you need to just refer to again. Here we are discussing two autosomal recessive disorder. What are autosomal recessive disorder? Autosomal recessive disorder are carried on the chromosome which are autosomes. This is a recessive disorder, which means that you need to have both the recessive alleles for the disease to show itself. If you have a single recessive allele, then you will be the carrier of the disease. If you have both the dominant alleles, then you will be a normal and you won't be a carrier. Sickle cell anemia. In sickle cell anemia, there is a beta globin chain which is coded by HBE allele. This is normal. If this is normal, at the sixth position, there is an amino acid glue coded for CTT. Then, if a person is having allele HBS, in this person, the CTT codon will be changed to CAT, and CAT will cause another amino acid BAL to be inserted into the primary structure of the sequence. So the primary sequence changes because instead of GLU, you have BAL, and the protein will fold abnormally, and that's why people have sickle cell anemia. Then we come to the disease with the albinos. People who are albinos suffer from albinism. Albinism is when the production of melanin does not occur in the body. And where does melanin occur? Melanin basically is synthesized by melanocytes, and these are the cells in your skin and in the base of your hair as well. And these melanocytes, they have an organelle called melanosome. Melanosomes carry an enzyme tyrosinase. Tyrosinase catalyze this sequence of reaction in which amino acid tyrosine in your diet, it is converted to dopamine, dopamine converted to dopaquinone, dopaquinone turns to melanin, and melanin gives the skin and the hair its color. In albinos, tyrosinase gene is not working properly, and the tyrosinase enzyme cannot be functional. That's why whatever tyrosine amino acid that they may be having, it can't be actually converted to melanin. So albinism and sickle cell anemia, they both are autosomal recessive disorder. When the parents are the carrier of the same recessive autosomal trait, there is 1 in 4 chance that the offspring will have the homozygous recessive disorder. There will be 2 in 4 chances that the person will be a carrier, and there will be 1 in 4 chance that the person will be normal. Not all disorders are autosomal recessive. A few of them can be autosomal dominant as well. Very few examples exist for autosomal dominant disorder. Here, the person who's having this disorder will have the dominant allele. So the disease is the dominant allele and not the recessive allele. For the person to be normal, for this case, the person must have both the recessive allele. The example is Huntington disease. The Huntington protein. Huntington protein is coded for uh, certain sequence, coded by a certain sequence from chromosome 4, and that sequence is having a small number of nucleotide sequence CAG. We are normal for Huntington protein because we have normal number of CAG. But people who have a mutation, who have the dominant allele, they have a stutter of this CAG. A stutter means that the several repeats of CAG will be there at the Huntington nucleotide sequence. So CAG stutter will be there. The more CAG stutter will be there, the earlier the condition appears. What conditions do we see in Huntington disease? The person will have neurological disorder. The person will suffer from chorea, which is flinging movement of the limbs. Mental retardation and progressive mental deterioration occur. And the people who normally diagnosed with this, they are diagnosed with by mid 20s, and they die by mid 30s. When parents are the carriers, these are the carriers of the same trait, you can see that there will be just 1 in 4 chance that the person will be normal. There will be 3 in 4 chances that the dominant allele will be passed on, and the offsprings will be suffering from the disease. That's it for now. Thank you.